So everything I've talked about up to now has been focused on secreted proteins. Those are soluble proteins that are eventually dumped outside the cell. And what I want to do is, even though many of those rules also apply to transmembrane proteins, what I wanted to do is kind of give you a sense of the diversity and that of the different types of membrane protein topologies, how they can insert into the membrane, and then really focus on the experiments that can be used to determine how a protein loops in and out of the membrane and kind of what rules determine the topology. Now, a lot of textbooks and a lot of classes go through a lot of detail about the various kinds of sequences that can determine membrane protein topology. I'm not really going to go into that because, in my opinion, the key thing is to understand how you can figure it out and kind of the underlying logic, because then it's easier to create exam questions, in my opinion, that actually test knowledge as opposed to your ability to spew back huge amounts of detail. So just to give you a sense of kind of how diverse transmembrane proteins can be. So here's the membrane. We can have a transmembrane protein where the amino terminus is inside the lumen and the carboxy terminus is outside. So this is the lumen and this is the cytoplasm. Conversely, here's the transmembrane sequence. Conversely, you can also have topologies where everything is flipped. And then you can even have things where the amino terminus is the actual transmembrane domain itself, and it's anchored in the membrane. So those are some simple things, but then you can have multiple membrane spanning proteins. So that's a protein that spans it multiple times. And these can get quite complicated. So for example, CFTR, the gene that is responsible for cystic fibrosis, it has 12 different membrane spanning domains. So it loops back and forth across the membrane 12 different times. And in fact, that topology was somewhat controversial for many years. And it took a lot of experimental work to figure out exactly which parts of the protein are on which sides of the membrane, which is really important for understanding how different disease-causing mutations actually work. And these are just the simple proteins that I'm diagramming here. You can also have other types of linkages, and in this case, I'm drawing one that has a GPI linkage, which is a completely different mechanism for anchoring something in the membrane. So these can get quite complex. And what I wanted to focus on, rather than kind of a lot of the sequences that determine how the different transmembrane domains get read out by the Sec61 channel, because a lot of that is like still relatively poorly understood, what I wanted to focus on are the experiments or the assays to determine membrane topology. And by topology, I mean the nature in which it loops back and forth across the membrane. So the first classic assay that people had was proteolysis. So you can imagine you have a membrane protein, and let's just put this on the cytosol. This is the lumen, and it loops back and forth, right? Now, if you treat that with a protease, what happens is the protease is out here on the outside, like a little Pac-Man coming in to chew up the external domains. And so what you end up with is just the fragments that are protected from the protease. So a simple way, let's see if I've drawn this correctly. So all these external domains, which I'm going to just draw in, these were all chewed up by the protease. And so what you have are 1, 2, 3, 4 different protein fragments. And so if you have antibodies to different parts of the protein, you can begin to do this kind of molecular Sudoku puzzle of trying to figure out, well, when I treat with a protease, what fragments do I have remaining? And what does that mean in terms of which parts of the protein are exposed to the cytosolic side and which parts of the protein are on the luminal side? So that's a classic way of figuring out by protease accessibility or sensitivity. And it's, it's very time consuming. It requires a lot of reagents, and it's difficult to kind of go from the pattern of bands that you see and work your way back. But it is a classic way of doing this. Now, the second type of assay would involve tagging the protein with an enzyme. And I'll show you why that's useful. And for this example, I'm going to use alkaline phosphatase. Which I'm going to abbreviate AP. You'll see in the literature and other textbooks that people use other types of enzymes, beta-galactosidase. Basically, any enzyme that has a substrate or an artificial substrate that can be turned into some sort of, from clear to some sort of color or some sort of fluorescent material. So anything that's an easy visual assay for enzyme activity. Now, why do you want to do something like that? So what people do is they say, okay, we've cloned this enzyme. We have alkaline phosphatase enzyme, and we have our protein of interest, right? And it has, let's say, 3 believed to be transmembrane domains, right? And at the C terminus, they're going to stick alkaline phosphatase. Now, this is a gene fusion, so basically you make your protein and then you remove the stop codon and you drop in alkaline phosphatase. So it's one continuous polypeptide. It's your protein of interest, and at the C terminus you have alkaline phosphatase. So you could imagine 2 different scenarios for this protein if you were to do in vitro translocation. Scenario 1 is the amino terminus is on the outside. It loops back and forth, and the alkaline phosphatase at the C terminus ends up on the inside. So that's scenario 1. Scenario 2, I'll just make it a different color to make it clear, is that the amino terminus is on the inside, and it loops back and forth 3 times because there's 3 transmembrane domains, and the alkaline phosphatase ends up on the outside. So these are 2 possible topologies of this 3 transmembrane domain protein. However, in scenario 1, so let me just make this scenario 2, make it clear. Scenario 1, the alkaline phosphatase is inside the vesicle. So what does that mean? It means there's no access to substrate. So let me just make this, so if the substrate's all floating around out here, alkaline phosphatase can't act on it and change it to a color. So everything remains clear. So that's scenario 1. So what happens with scenario 2? So with scenario 2, what happens is the alkaline phosphatase is outside the membrane. And what that means is now the enzyme substrate can interact with the enzyme, and let's just say when it interacts with the enzyme, all of a sudden it starts turning or generating a blue color. So the substrate changes color. And so you can tell based on where the alkaline phosphatase ends up, whether it's inside or outside, and if you have an idea of how many transmembrane domains you have in the protein, you can figure out the topology based on where the C terminus ends up. So those are the assays, and I use them in a lot of different problem sets in terms of figuring out, like, given a specific scenario or a set of experimental results, can you figure out what the actual topology of the protein is? But there are some standard rules on how the membranes insert or organize within the membrane. And so I'm going to walk you through a few of those just to give you some context. Some of these are just logical implications of how membrane proteins traverse the membrane, but it's always good to just like draw them out into the open so it's very clear. So the first thing, and this is really useful, is if you have an odd number of transmembrane, and I'm abbreviating that TM, transmembrane domains, then the amino and carboxy terminus, the ends of the protein, are on opposite sides of the membrane. And you can kind of see that here. So if I drew a membrane, let's just extend it out a little bit. So if I go 1, 2, 3, N and C are on opposite sides. And if I go, I can start on the opposite side. 1, 2, 3, 4, 5. Pretty much no matter what I do, and it's just baked into, if you cross the membrane 3 times, you end up on the opposite side of where you started. So conversely, an even number of transmembrane domains, then the amino and carboxy ends are on the same side of the membrane. So what does that mean? Well, that means that's one of the reasons that the alkaline phosphatase assay is so powerful, is that if I know how many transmembrane domains there are, or I have a good idea, and I know where the C terminus is, because based on if the alkaline phosphatase is inside the membrane or outside the vesicle, then I can essentially draw the entire molecule and how it spans the membrane. So that's why this particular rule, it's more an observation, and I'll just quickly draw an even number just to give you a sense. 1, 2, 3, 4, 5, 6. Give you a sense of how this rule applies. So it's more just a kind of a mathematical observation of how odd and even crossings work. It's almost just a basic rule of topology. But when you start combining that with the experimental assays, it becomes very powerful. So second rule, and this is more an empirical observation, and so a lot of professors kind of key in on this because it's something, kind of a little bit of trivia that people like to play with, is that charged amino acids determine the orientation of the transmembrane domain. And what do I mean by that? It means that if you have positive charges, is always on the cytoplasmic or cytosolic phase. So what does that mean? So obviously a protein has a bunch of, most proteins that are going to have hydrophobic residues where the transmembrane domain is, because it has to stay embedded in the membrane. But then you can have, a variety of polar, nonpolar, positive, negatively charged residues. But the observation is that if you look at a protein once it's already embedded in the membrane, so here's the membrane, I'm just going to draw it with a different color. So if you imagine this, and this is cytosolic, and this is luminal, if you were to guess where you might see a cluster of positive charges, those positive charges would be found on the cytoplasmic side of the transmembrane domain. So if you were to draw this out as a linear sequence, so assuming that this is N and this is C, what you would see is, let me just, so this is 4 transmembrane domains, 1, 2, 3, 4. I might expect to see positive charges could be here, a cluster of them. They don't have to be. That's always the thing. It's not something you always see. But if you saw a cluster of positively charged amino acids on one side of a transmembrane domain, those are almost always found on the cytosolic face. So I could see them. Let's see. No, that would be here. So here, let me back that up. So if you follow this around, so you start on the amino terminal side, this is cytosolic, this is lumen, cytosolic, lumen, cytosolic. So you can eventually see, oh, I'm going to see some positively charges maybe here, and then maybe on the far side of that one. So if someone gives you a sequence and says that there's a cluster of positive charges on this side of a transmembrane domain, where would you expect it to be? You would expect that that cluster would be facing the cytoplasmic side. So it's a useful trick. It's also something a lot of professors put into problem sets and various things. So it's a good factoid to know. And then lastly, I'm not going to get into the details of these sequences. I'm just going to let you know that they exist. But in order to generate these complicated membrane protein topologies where they span and loop multiple times, as you might imagine, figuring out how to spool the spaghetti so that it loops across the membrane 12 times with the correct portions on the correct side of the membrane, it's a little bit challenging. So there's a whole set of what are called topogenic sequences. And they're relatively poorly understood, but they're the ones that instruct the Sec61 channel what to do, like to continue translocation, to stop translocation, to allow that transmembrane domain to exit sideways and to diffuse laterally into the membrane. So, there's a lot of information content, but they're poorly understood. And eventually, hopefully, we'll understand them well enough that when given a primary amino acid sequence, you could predict the topology of the protein with a high fidelity. But 2 sequences that are just worth knowing about their existence because they often crop up are signal anchor sequences and stop transfer anchor sequences. So why mention these? Signal anchor sequences instruct the transmembrane domain stays in the channel while translating. So what that allows it to do is, like, if you have a huge amount of sequence after the transmembrane domain, you don't want that transmembrane domain to continue on. You don't want it to spool through the channel. And if you let it go lateral, it might start to thread into the channel and create an additional fold. So what often happens is that transmembrane domain, when it has a signal anchor sequence, it anchors it, and then the ribosome essentially detaches from the channel and just continues translating the rest of the cytoplasmic domain. In contrast, these stop transfer anchor sequences instruct the transmembrane domain to exit the channel. And that's really important. So if you're making 12 different transmembrane spanning regions, so at a certain point, you can't cram 12 different transmembrane spanning peptides into the channel. So there comes a point where you do it, you have the transmembrane domain in the channel, but when you're queuing up and you're ready to move on to the next transmembrane domain, this stop transfer anchor sequence allows it to traverse laterally, and then it can start working on the next set of loops. So those are both really important topogenic sequences. Again, I'm not going to go into it, but it's important for you to understand that there are these signals that tell it how to loop in and out, and they're all being read by the channel.